Good afternoon and welcome to the American Floral Endowment Grow Pro webinar series. I'm your moderator, Melissa Muñoz. I'm a member of the American Floral Endowment Young Professionals Council, and I'm an assistant professor on small fruit crops horticulture at Clemson University. Today's session is on Ruh Roh and Managing Oomycete Pathogens in Floriculture Production. On behalf of the endowment, I'm excited to be part of the AFE Grow Pro webinar series that features a new topic every month presented by an industry expert. The webinars are free to everyone thanks to the generous support of AFE sponsors. The views and opinions expressed in the webinars are those of the speakers and do not necessarily reflect the views or positions of the AFE. As an independent nonprofit, the AFE cannot endorse any specific product or opinion. If you would like to learn more about our webinar series or are interested in becoming a sponsor for a topic, you can find more information on the AFE website at endowment.org/growpro, and you will find the link in the chat. Today's session was pre-recorded in English by Dr. Joana del Castillo Munera. After the presentation, she will join us for a Q&A, so please feel free to submit your questions through the Q&A feature or the chat at any time during the presentation. We will answer as many questions as we can before the end of the hour. Any unanswered questions may be answered through a separate email exchange. This session is being recorded and will be shared through AFE's YouTube account. Through YouTube accessibility features, you can access closed caption in other languages. To get us started, I would like to share a bit about today's expert speaker. Dr. Joana del Castillo Munera is an assistant professor of cooperative extension at the University of California, Davis. Her main interest as plant pathology center on the basic biological understanding of fungal and oomycete pathogens, and on the development of applied research to solve current problems that greenhouse and nursery industries face. She is particularly interested in early pathogen detection, developing and improving cultural practices based on risks of pathogen exposure, and finding sustainable solutions for disease control through studies of fungal and oomycete biology, population genetics, and community ecology. Joana, welcome and thank you for presenting today on oomycetes.
Good morning, everyone. Thanks so much, Melissa, for the introduction. It's a pleasure to be a seminar speaker in the American Floral Endowment seminar series. This is my first time participating on it, and I will be talking today about root rot diseases of oomycete pathogens, which are pretty frequent in floriculture production. Before I start talking about what are oomycetes, I want to start into why do we care about these organisms in the floriculture context. So first, due to a quality issue, so plants in the greenhouse or nursery can look asymptomatic, so they can look pretty healthy, they can look with pretty producing flowers, but that asymptomatic plant can be infected. And then once the plant leaves the nursery facility, it can die after it's transplanted in the landscape, or it can die at the retail store. And so why that can happen? So when we have the plants in the nursery, usually they are grown under pretty good conditions. They have all the water they need, they have all the nutrients they need. But once they leave the nursery, say they are into the landscape, they are into the homeowner, they can experience some plant stress. So what are these type of plant stress that the plants can experience? They can experience drought. So for instance, if the plant is not getting as much water as it should get, is, for instance, also think if you are at a restoration site where there's not irrigation provided, or if it's staying at a landscape where there's not much water, or for some reason the person in charge of the irrigation forgets to irrigate it, or at the retail store the plant is not watered. Or for instance, if the plant is transplanted and is a lot of standing water in the ground, or there's like heavy rains by the time the plants were transplanted, these plants can undergo stress. And once we have this type of stress, in this example, we have drought or saturation or overwatering, is when the plants start exhibiting disease symptoms. What are the type of symptoms? We'll be seeing a lot of those in this presentation. But for instance, in this picture, we have a dark pomegranate crop, and we can start seeing like some defoliation, we can start seeing some wilting, and then suddenly we can see some plant death. Continue with the quality issue, we can have pretty healthy asymptomatic looking plants. This is the case of a bay laurel that we had in the greenhouse experimental place here at UC Davis, that we artificially inoculated with a Phytophthora species. And after a year that this plant was infected, we didn't see any symptom at all, but we know the pathogen were present. And so we then sample these plants, and effectively we recover these pathogens. Not only Phytophthora species of that are non-regulated can be affected, but also asymptomatic plants can harbor quarantine pathogens. For instance, here in California or in the Pacific Northwest, in the early 2000s, we have this sudden oak death that was caused by Phytophthora ramorum, which is a regulatory issue, and nurseries are pretty heavily regulated to avoid the presence of this particular species, as it can result in quarantine. So, of course, we don't want those quarantine pathogens present in the nurseries. Another reason why we care about these organisms is that it can cause yield loss of your crop. This is an example of a cistus crop, and the two plants you have in here, as you can see, are completely dead. Of course, these plants cannot be sold. These plants cannot leave the nursery, so they will die in the nursery. And then an important thing, once these healthy plants become infected with Phytophthora or Pythium, there's no cure for infected plants. Once the pathogen is there are not treatments that are gonna kill or completely eliminate the pathogen of that. And also, if we have infected plants in the nursery, as the case of these ones, they can be a source of inoculum, like they can harbor the spores or the pathogens, and then they can get into healthy plants and spread the infection in the nursery. Continuing with pathogen spread, not only while the plants are in the nursery, but once they leave the nursery, is that if we sell infected plants and then they are transplanted somewhere else, this pathogen can be spread. For instance, here in California, we had cases where we have native ecosystem restoration sites that are sensitive habitats for native plants that have, we call it failed restoration projects. This is an example of some manzanitas, we are native Californians, that are completely affected. As you can see, all this defoliation, all this plant dead by infected plants. And so it's really important to prevent the introduction of these devastating Phytophthora species into native habitats. And then, if not necessarily into restoration sites of native habitats, but for instance, a garden. Recently, we were called to visit a garden that was installed or set together at a hospital. And they were concerned, the landscape architect was concerned because all the plants that were planted were dying. Most of these plants were native California plants. And then we sampled the plants, and then we recovered several Phytophthora species for this landscape project. So definitely it can be a concern to spread these pathogens out of the nursery into native sites or in general landscapes in a city. So now that we talk about why do we care or the importance of these organisms, let's talk some of the biology of it. So in biology, we like to use these life trees. This is the one that you can see in the picture, to have a better perspective of like how the organisms are closely related, and how by this we can have better tools to study their biology, understand them in our case, in the agriculture side, how can we propose management practices based on the type of biology that these organisms have. So the oomycetes have pretty similar characteristics and ecological traits that fungi or true fungi have. But if we look at the fungi are grouped in this group of the tree of life. The oomycetes, by many molecular and DNA studies, happen to not be that closely related to true fungi. They are more closely related to brown algae. They're in a group known as Straminopiles. One important characteristic of these organisms is that they have spores, and these spores have two flagella. And so they have a hairy flagella and they have a hairless flagella that allows them movement. The name that we have for these spores are called zoospores. And the reason for them to have these two, or one of the traits that they're conferred by having these two flagella, is that they can swim and reproduce through water. If we compare that with true fungi, they do not have that. There's a very early lineage of fungi that only have one flagella, but then by evolutionary purposes, that flagella was lost, and then the true fungi spores don't have the ability to move and swim as the oomycetes that are more closely to brown algae has. And so Phytophthora and Pythium, that I'm sure you have heard that may be present in infected crops, they are an example of this organism, more closely related to brown algae. And this is an example, if we look at these organisms under the microscope, how do they look? So what you see in this picture is a sac, and this sac is called an sporangia. If you see inside this sac, there are all these like brown blobs. And then if we look closer into these blobs, these are actually zoospores. So as you can see, the zoospore is the circular structure that has a flagella, and that's what allows mobility. As they have these traits and they're able to move, an important component for them to spread is water. They really like water because they can swim and reproduce in water and propagate through that. And as we'll be seeing that in our presentation, water is a pretty important component for an adequate management of these organisms. Now let's start talking about Phytophthora. Phytophthora species are well-known pathogens of thousands of ornamental plants worldwide. They are distributed everywhere. What you see in the right are some examples of the different sacs or sporangia or other reproductive structures of these organisms. They have been reported to cause over 60% of fruit diseases and 90% of crop diseases in woody ornamentals. An example here, we have a root infected with Phytophthora, and then how can we determine if a root is infected by this? This is in the plant that was brought to the laboratory. And then if we start seeing some browning of the roots, or we call it brown or root discoloration, is where we see that there's a highly chance that these roots are infected by a root rot pathogen. We have, of course, to do further testing in the lab, isolate the organism, and identify to make sure it's a Phytophthora, but this is an example of an infected plant from which we recover Phytophthora. To date, there are over 200 Phytophthora species described. And of those, there are over 50 species that have been reported as plant pathogens of annual, perennials, herbaceous, shrubs, and ornamental trees. The picture we have in the right is an example of a rosemary crop. You can start seeing yourself what do you see unusual from this crop or what doesn't look quite healthy. And so we have some yellowing or browning of the leaves. So we also call it necrosis when we see like these brown leaves or brown branches. We call branch necrosis. From this crop, we recover a Phytophthora species, And another example that we have here is a wax flower that you could see is pretty, there's a thinning, so it's like pretty defoliated, all the leaves have fallen off, and from this one we recover Phytophthora species as well. I'm going to show you here more examples of plants infected with Phytophthora and how do they look like. This is an example of gardenias, what we could see, necrosis of some of the leaves. Some of these plants had a stunting, or they're smaller compared with healthy looking plants. And an important factor of these diseases is that we get to experience these symptoms on the ground, or sorry, above the ground on the foliage. For instance, like this, like wilting or necrosis or defoliation. But this pathogen is not present on the leaves or not present on the foliage. These pathogens are actually present on the roots. So if you're in your facility and you're experiencing some of these symptoms, a recommendation I have is to toss the plant and go over and look the roots. And so in this picture here, we can have an example of browning of the roots. And then also what I recommend is like looking how the roots of a healthy plant look like. And usually a healthy plant looks like with more like wide, healthy roots, and the root is covering a higher portion of the substrate. And so infected plants usually have these like reduced root biomass as an indicator of disease. Another example of an infected plant is the wax flower that I showed in the previous picture. As you can see, we have like this like thinning and defoliation. And then if we look from here, you can see the crown that was with this like brown discoloration indicating the disease. Now, let's go over the life cycle of this organism. And so let's start with the zoospore. So let's start with one of the infected roots that you have in the previous picture. These roots mainly would have these zoospores. Remember, these zoospores are the structures that can swim and move through water. And so we have these zoospores that leave the root. In some cases, they can form cysts, and when there are ideal conditions, such as ideal humidity, the host is present, they can germinate, and they germinate creating this like hyphae or mycelium that for us is the body of the oomycete, and these mycelia can produce sporangia. The sporangia, as I mentioned in the earlier slides, are these sacs that contains all the zoospores. And then if they have good water conditions, they can keep dispersing through water, and this cycle can repeat over and over again. Besides infected plants through zoospores, they also have resisting structures. In the case if they undergo sexual reproduction, there's an oogonium that can fuse with an antheridia, and then that gives rise to an oospore. These oospores, if you can see in this scheme, has many cell walls, and by having these many cell walls is a structure that can be resistant and survive for a long period of time. So there are studies that have identified survival of Phytophthora over years in the soil. And then after sexual reproduction takes place, again, mycelia is formed, sporangias are formed, and zoospores are keep forming. And then disease goes over and over infected plants. If we have, in the case of asexual reproduction, there are also resistant structures that are called chlamydospores, and these chlamydospores also can overwinter and survive in the soil for a long period of time. Now, what are environmental conditions that favor these Phytophthora infections? So one is humidity. As I mentioned, water management is pretty important. Another one is temperature. So usually cool to warm temperatures favor the presence of these pathogens. The optimal temperature range depends on the species that is infecting your crop. But depending on the species I mentioned, can be between 59 to 75 Fahrenheit. Soil and substrate, if there are, again, as I mentioned, saturated soils, if there's a standing water in the ground, if there's high clay content, those favor the presence of these organisms. And then if your source of water is contaminated, of course, that can keep spreading the pathogen. And then besides considering environmental conditions that favor disease, also you should consider the cultural practices that are in place that can facilitate the spread of these pathogens. So if non-adequate cultural practices are in place, that also can help to spread this pathogen. Now we will move with the other group of organisms, Pythium. I'm using two more words, sensulato. Sensulato means a broad group of Pythium. The reason for that is that because we usually knew this Pythium as only one genus, and I will go in details later, but now it is split into 5 different genuses. And so there are over 200 species described as plant pathogens. This can be general necrotrophs, as meaning like they can infect organic matter in the composition, or they can infect plants that already have some type of root rot. They can be pioneer colonizers, so they can be primary colonizers of these tissues. They have a pretty wide host range, and usually they can attack fine roots of young plants. So an example of a young plant in here, this is a geranium seedling that it was heavily devastated by Pythiums. And so they can cause pre and post damping off. They affect the embryo, hypocotyl, and emerging radicle, and they cause stunting, chlorosis, and damping off and death of many seedlings. On mature plants, they can cause reduction of root biomass, and as a consequence of that, the plant cannot uptake enough water and nutrients. And then you will see all the wilting, in this case of this geranium, of this, poinsettia, mature poinsettia. In some cases, if you compare with other healthy looking plants, it can look stunted. But again, it's because there are not enough roots in this plant in order for them to uptake nutrients and uptake water. More example of this disease. This is a native California pine, Pinus saviniana, that when we collected them at the nursery, they were stunted compared with other seedlings there. And then in the lab, we went ahead and looked at the roots. And then in this next picture, you could see that we were seeing pretty clear this browning of the roots or root discoloration. That is an indicator of a root rot pathogen being present in the crop. This is an example of some infected plants that we did an experiment in the lab. These are native sticky monkey flowers, native here California, where we have a healthy plant that was non-inoculated. And as you can see, how stunted and devastated look this one, and this seedling that is completely dead after artificially infected with a Pythium species. If we look at the roots of these plants, the healthy, non-inoculated plant look with a pretty strong root, and like you can see, the root biomass is significantly higher if we compare with these little seedlings that are completely affected by Pythium, where their roots did not progress at all because this pathogen significantly affect all the roots of this plant. So as I mentioned earlier, in the Pythium sensulato group, and so in recent studies based on the DNA of these organisms, it was determined that Pythium is not one genus, but is divided in 5. One of those is, Phytopythium, then Pilasporangium, Elogisporangium, and Globisporangium. So for instance, there's a well-known Pythium former Pythium that was called Pythium irregulare, that now the name changed to Globisporangium irregulare. So when diagnosing diseases, usually this will be determined as Pythium root rot. But so, if you get a diagnostic result and they say, for example, globis perambium irregulare, we are still referring as what was known as Pythium. In terms of the ecology of these organisms, they survive in the soil as oospores, the main structures. Also, we now have something known as hyphal swelling, which is actually hyphae that have swelling and that can confer, as I mentioned, these overwinter capacities that they can survive in the substrate or in the soil for years. For the propagules to germinate, they get stimulated by root at seed exudates and also organic matter that stimulates the oospore and sporangia germination. And the zoospores formation are stimulated by high moisture content. And then the zoospores also are attracted to germination seeds or roots and in presence of water. As I have mentioned since the beginning of the presentation, water is a pretty important source for this pathogen mobility. Now, environmental conditions that favor Pythium infection. What do you guys think may be the first condition? Answer that to yourselves. So definitely humidity. High saturated soils favors the presence of this pathogen. Not having good drainage or bad drainage that can prevent that from happening, of course, favors that. Temperature, it depends on the species. Usually it's a range from 50 centigrades to 30 centigrades, and then that definitely can affect the pathogen from infecting a plant. We have done experiments in our greenhouse where we started at higher temperatures, say like at 25 centigrade degrees. This is in a case for a Pythium species inoculating brassica seedlings, and then we didn't see any disease symptom development at temperatures at 24 or 25 centigrades. But then when we were under cooler conditions that were like under 18 to 20 centigrade degrees, we could see how this Pythium pathogen caused significantly reduction on the root biomass and stunting of some of the brassica crops. So definitely pathogens can be present there, but if there are not the ideal environmental conditions and the host present, disease doesn't take place. And also, if the source of irrigation is contaminated irrigation water that can contain these pathogens, that's how they can be spread into the nursery. And again, definitely practices in the nursery can be conducive for this pathogen, if not best and sanitation practices are in place to prevent the spread of these organisms. So what to do if you think you have this pathogen in your nurseries? One thing we recommend is contacting your extension agent or farm advisor present in your area to visit your facility, take a look at some plants and see what can be going on if you're seeing something unusual with your crop. Consult university extension materials. There are many publicly available resources produced by the universities that are accurate and reliable into how to identify if you have these diseases. But the most important thing is if you're not certain if you have these organisms, is to have an actual disease diagnostics. And so you can contact diagnostics laboratories present in your state, in your area. The link that you see below is the National Plant Diagnostic Network. This National Plant Diagnostic Network has laboratory diagnostics in every state, so you can access them and finding them there to see where you could send your samples. And having an accurate disease diagnostics is the very first step to actually propose an adequate disease management program. So for instance, like if to manage oomycetes, you are applying fungicides, as I mentioned, oomycetes are not fungi. So the active ingredient of the fungicide you may be applying is not effective for your oomycetes. And so you want to be using some chemistries that are effective against oomycete instead of fungi. So it's just an example of how by having an accurate diagnostics, you can actually better manage the pathogens present in your facility. So Now, in the next part of the talk, we're gonna see if we think we have these pathogens in the nursery, first we need to think where these pathogens can be coming from, and then how can we manage them. And so start thinking to yourself, where can these pathogens be present at each of your facilities? And also we will have these sources as nursery hazards, and we're going to minute why we think of nursery hazards. So think of new plant material or propagules coming to your nursery. If they come from other nursery that you don't know the practices in place there. If there's contaminated ground, and as I mentioned, these organisms have overwinter structures that they can survive over years. And then think if your plants are in direct contact with this ground. What is your source of water? If your source of water is contaminated with these zoospores while you're irrigating, are you also inoculating your crop with these pathogens? Use containers. Do you reuse containers in your facility? If so, do you clean and sanitize these containers? And then contaminated potting media. What is your source of potting media or substrate? So in order to manage these hazards, in 2012, Jennifer Park and Nick Grunwald at Oregon State University came up with a risk assessment program for nursery production. And then basically the proposal for them is how to implement a systems approach for disease management. And so an example of this system approach is the implementation of a hazard analysis of critical control points, or HACCP program. This is widely used in the food industry, say in restaurants and in food production. And the general goal of this, for instance, for food production, is prevent foodborne diseases. In our case, is like how can we prevent plant diseases in the nursery? And so the main components of this program is identify the hazards, which we have done it in the previous slide, and then identify the critical control points, or CCP. These are the steps of processes in the nursery production that may pose a higher risk for disease infection. Once these critical control points are identified, they can be corrected. How can they be corrected? Implementing best management practices to prevent or reduce the infection risk of the pathogen. So let's start with the first hazard. So we have new plant material coming to the nursery. And so if you bring plant materials from other facilities, it's really important that you know which practices are in place at the nursery. So if possible, buy plants from certified nurseries. If you are the one who produce 100% of the crop, as like if you collect propagules or you collect seeds for production, make sure you have, since collection of the seeds or collection of the propagules, best management practices in place to prevent the introduction of pathogens in the nursery. If you have plants coming from another nursery, it's important to scout the new plant material. Look if there are some symptoms. Look not only at the foliage, but look at the roots. Ideally, place this new incoming plant material in quarantine area so you can keep monitoring them and preventing and looking for any symptom development. Ideally, if possible, if you can test some of the new incoming plant material to guarantee your plant is free of pathogens. And then what is highly recommended is do not mix incoming plant material with infected material. The reason for that is that you may be growing pretty healthy crops and you bring uncontaminated crop, and then you're spreading a new pathogen that came from the new plant material, and you're infecting your crop. So that's something that is ideal to prevent. Following up with the nursery hazards identified, if there's contaminated ground, a critical control point is if there's direct contact between the soil and containers, as you can see in that picture. Best management practices. If it's feasible, we recommend using raised benches, as the raised benches prevent direct contact from the plants and the ground. If that's not feasible, at least it can be at gravel, rock barriers, or landscape fabric to prevent the direct contact among the plants and the ground. The picture of the raised benches that you're seeing was taken from a native California nursery here in California that is part of a program that is called Accreditation to Improve Restoration. This is the website of our program. This program was initiated as a response to some of these failed restoration projects that we had here in California. And the main component of this program is to implement a systems approach to exclude Phytophthora of nursery production. We have strict best management practices that you can consult in this website, and success of the practices we propose is we have tested over 37,000 plants, and we have not detected any Phytophthora from these plants. In this program, we currently have 24 participating nurseries, with 10 nurseries fully certificate and some other ones in process of getting accreditation. So if you're interested in learning more of some strict best management practices, you can go into our website to look into those. Moving on with the more critical control points in contaminated ground is that the splash dispersal of pathogens from contaminated ground. So for instance, if there's presence of standing water and then you're irrigating and there's a splash dispersion, by this splash dispersal, zoospores can be moved from the ground to the healthy plants. And so important for that, prevent standing water, avoid overwatering, improve drainage, and again, having raised plants not in direct contact with the ground will prevent that from happening. Also, if there are Phytophthora or Pythium infections, and then it's probably that the substrate or the soil that these plants are infected. And so if you work with tools, equipment, stuff, they can be a source of pathogen dispersal too. And so the best management practices is to clean any equipment. It's important to clean first. So it's basically removing any soil, removing any organic matter, and then apply a sanitizer, because sanitizers are effective, but they can reduce efficacy if they're in presence of organic matter. So before doing any sanitation, it's really important to clean first and remove any debris or soil. And this goes for shovels, it goes for shoes, it goes for vehicles. And so make sure to disinfect these materials when moving from a contaminated area to a clean area. Or ideally, if you know an area is contaminated, work with that at the end of your day and start by working in areas with clean production and then moving forward to various production in order to reduce the risk of pathogen spread. Also, the soil can be contaminated by infested foliage. So for instance, like if there's plant debris on the ground that can harbor inoculum. So what we recommend is prevent and remove any leafy debris from accumulating on the soil. And then now we'll be moving into our next hazard, which is contaminated irrigation water. So if your source of water is surface water, such as pond, river, stream, or if you collect the runoff water and reuse it for irrigation, we call that recycled water, there can be a risk of this water being infected with Phytophthora and Pythium pathogens. There are several studies out there that show proof of that. And so the best management practices would be use well water or municipal water that is free of this pathogen or this infested water. If you suspect your water may be infected with Phytophthora, we recommend testing the water for that. You can reach out to the diagnostic lab in your state or research universities that work with these organisms to get your water tested. So how the testing is done is that we deploy baits. The picture that you see there is an example of a rhododendron leaf that was deployed in a pond. And then all the brown spots that you see around are lesions caused by Phytophthora that are an indicator of that this species was present there. Continue with another critical control point in irrigation water is if there's a splash dispersal of pathogen, again, if there's contamination of waterways. Best management practices: have good irrigation practices, prevent standard water, have well drainage, avoid over irrigation, again, use raised containers off the ground. If possible, schedule water in the morning so the plants can and the substrate and the soil can dry out through the day. And then collect any runoff water and disinfect it if that is gonna be reused or recycled. Moving on with the fourth hazard, this is use containers. And so we are aware many nurseries reuse their containers over seasons. The best practice is, well, one, don't do that and use new containers. We know in some cases that's not feasible. But so then clean before mechanically remove any soil debris, any plant debris, any organic matter left, and then sanitize these containers. The most effective measure to sanitize and to actually kill Pythium and Phytophthora is heat treatment. And so if the reused containers are exposed to heat moisture, they can kill Pythium and Phytophthora species in temperatures above 110 degrees, and then true fungi in temperatures above 140, 150 degrees. So some examples of container disinfection method, as I mentioned, the most effective one is aerated steam. What we recommend is that it reach a minimum temperature of 140 Fahrenheit, and then it's not only reaching that temperature, but the duration of that temperature. So it's like we require at least 30 continuous minutes to actually at that high temperature to kill pathogenic spores. And then going over the last critical control point is use of contaminated media. The best management practices of that, if possible, use certified media that can be free of pathogens, or steam sterilize the media to prevent or to reassure that no pathogenic spores are present there. If also media storage is important, store it in an area free of contamination, away infested water, soil, or plants, and make sure to use clean equipment to mix the planting media. So to finish, do nursery condition favor oomycete presence? Ask that question for yourself. The answer is yes, if there are no best management practices in place to prevent the pathogen infection and dispersal. And then, as I mentioned, it's important to consider all the steps and processes in plant production that can pose a greater risk for disease infection or for pathogen spread in order to prevent that from happening. And then implement best management practices in these critical steps to reduce the likelihood of disease. And with that, I would like to thank you all for listening, for the organizers to invite me to be a guest lecturer, and I'll be happy to take any questions. This is the information from my website. And before I go and I'm ready to answer questions, I would like to announce the next seminar speaker that will be on February 17 about quality assessment of biological control agents by Dr. Raymond Cloyd from Kansas State University. And now with that, I'll be happy to take any questions that you may have. Thank you.
Thank you very much, Joanna. And I see that our audience is already engaged and we have several questions between the chat and the Q&A. So the first one is, can Phytophthora and Pythium also affect the leaves of a plant? And also, do you have experience with peroxyacetic acid to fight Pythium and Phytophthora?
Yeah, so I'll start with the first question. If Phytophthora and Pythiums can infect some leaves. So for the case of Phytophthora, some species, yes. A classic example is Phytophthora ramorum, the causal agent of the southern oak dead, that can infect the leaves and cause lesions of the leaf. Do we call this Phytophthora ramorum an aerial Phytophthora, as it can infect the foliage of the plants? There are some other species, for instance, like Phytophthora cryptoga, that besides infecting the roots, can also infect some leaves. In terms of pythiums, I have not seen any reports of pythium infected leaves. Pythiums are mainly root rot pathogens. And the next question is
Peroxyacetic acid.
parasitic acid. So something that we're currently working on. So as like you can see, we recommend, like sanitizing, using sanitizers. And next questions that we have is like, which sanitizers are the most effective? I have done some experiments with botrytis, not with Phytophthora or Pythium. And then like where I have found that the different sanitizers that we have tested all are very effective at eliminating botrytis if we compare with doing nothing or just adding water. So all sanitizers work, but for instance, in the experiments we have done in the lab for botrytis, we have seen that bleach and quaternary ammoniums have been the most effective treatments. With Phytophthora per se, definitely that's an area that we are currently interested in testing some of the sanitizers' efficacy against these organisms. So I hope by the end of the year we can have some results on that, as we have some incoming experiments related to that.
Awesome. Another question is, and it may be on part of what you have been testing, and also they may need to tune in for the coming webinar, but any biocontrol products that you find to be effective against oomycetes in organic cultivation in California?
That's a good question. So definitely there are biological controls available. The efficacy of the biological controls depends pretty much on the pathogen you're testing that against with and the environmental conditions that you have. So for instance, like in or specifically in California, we have started testing some Trichoderma hercianum, that root shield is the most common product. And then we really haven't seen a significant improvement if we compare with a synthetic fungicide. But definitely that's more research that should be done. And again, as I mentioned, the efficacy of these biological controls is very variable, depends on the environmental conditions that it has. And then what is ideal for the use of these biological controls is a preventative and not as a suppressive. So it's like the idea is like it is applied beforehand, before the pathogen comes. So the biological control has time to start colonizing, in this case, the root zone of the plant, and that can give more characteristics, more abilities for these biological controls to better counteract in case a pathogen comes later. But specific for California, we need to do more research on that.
The other question from Ben Harris here, can a wet wooden bench harbor disease?
That's a great question, and the answer is yes. So we actually have done some research. This is not with ornamentals, but with vegetable transplant production, where we are taking environmental samples of benches. And so we had benches that were made of wood and in very close contact with the ground. And then also we had benches that were made of stainless steel that were at least 60 centimeters from the ground. And the number of the microbes, not pathogens per se, but the number of microbes or microbial loads that we recover from wooden benches were significantly higher than that of stainless steel benches. So definitely wood benches can harbor pathogens and can harbor propagules. What we recommend is using some sort of like plastic or galvanized metal or stainless steel metal that, one, are easier to clean, and two, are less likely to harbor any pathogens, as the case of the wood benches.
Perfect. And we actually have kind of the same question from two different people regarding what kind of sanitizers do you advise also for cleaning equipment. And I know you already mentioned quaternary ammonium salts and bleach, but do you have any additional comments?
Yeah, so as I mentioned, some of the work that we have done comparing only sanitizers with efficacy, and this was done with botrytis, not with Phytophthora. And so we tried some, well, we tried bleach, we tried quaternary ammonium products, and we tried some parasitic acid products, and we found that again, all are effective at significantly removing, in our case, botrytis propagules from the surfaces we infected, as compared with doing nothing or adding water. But the ones that consistently were most effective were quaternary ammonium and bleach. One challenge with bleach is that it can be corrosive, so it can ruin like your tools. But for instance, like it can right after it's applied, it can say like 5 minutes after it can be rinsed with water if that can prevent corrosiveness.
So the next question is, I'm curious if you have a specific ORP target you will recommend to suppress the growth of pathogens.
What is an ORP? If the person can elaborate.
Let's see. And I think, Justin, could you specify oxidation reduction potential?
Oxidation reduction potential for
For irrigation water to suppress the growth of a pathogen.
oh, okay, so water treatment. So yeah,
Mhm.
that's another topic. So definitely there are some resources available online that have recommendation of water treatment methodologies. So for instance, one consists of like chlorine injections. And so it's like the chlorine injection is applied to sanitize their water. I have heard of other nurseries that use like copper treatments to treat their water. And I have not personally tested efficacy of different water treatments to eliminate pathogen loads, but I'm aware of there are some resources available. For instance, slow sand filtration is other one. Ozone water treatment is other option available.
Great. Another question that we have is, do you know of resistant crops or do these two attack all plants?
This is for Phytophthora.
I think Phytophthora and Pythium.
Yeah, that's a great question. So, um, as there are as many Phytophthora species or 50 Phytophthora species that has been documented to infect ornamental crops, as they have a wide diversity of crops in which they can infect. We cannot say per se there are these species that are resistant to Phytophthora. But for instance, we could see which there are reports of grasses. So it's like there are not many reports of grasses being infected by Phytophthora. So I would say like grasses would be a safe bet for preventing Phytophthora infection. And then within certain ornamental species grown, I could see there may be some tolerance depending on the cultivars that are grown. So for instance, we're doing some experiments now with lavender and Phytophthora nicotianae, and we saw like one cultivar was less susceptible than the other. So I would say like we can work around tolerance, but not around full resistance.
Perfect. Here in the Q&A we have, it's steam sterilization of raised beds an effective means of eradicating potting media of oospores and chlamydospores.
So definitely steam sterilization is the most effective treatment for eliminating Phytophthora or Pythium propagules. What should be considered is that the minimum temperature that should be reached is 140 Fahrenheit degrees for continuous 30 minutes if you have like a contained area that you want to steam sterilize.
Great. Also, do you have any frag codes that you recommend for Phytophthora and Pythium control?
I can look that. I don't have it right on my screen, but I can look at that. For instance, like some of the fungicides that are effective are, well, mefenoxam is a well-known fungicide that for years has been used for all mycete management. Um, There is also the oxyathiopamine that, like, for instance, like Orondis is the trade name, or Segovis is the trade name of one of those. That is some of the resin chemistries that have proven very effective for those ones. Also, there is some like phosphites that has been used for years, and those ones have shown efficacy to manage Phytophthora pathogens.
Sure. And also, kind of a follow-up question on the sanitizing products. How long you can reuse a sanitizer solution, let's say a peroxyacetic acid or a quaternarium ammonium solution?
Yeah, that's a great question. So it's important when you're using any of the sanitizers, read the label, use the concentrations that are recommended to be used, and also some of these sanitizers tend to lose efficacy while they're exposed to light or while they're exposed to heat. So it is recommended to change them pretty frequently. So for instance, like for food baths, if you start a food bath in the morning, what we recommend is that every day that food bath is replaced, because the active ingredient that is there can degrade with time. Also, if there's organic matter present, the sanitizers are going to be less effective.
And also, do you have any online resources that you recommend for exploring irrigation water sanitation?
Yes, we have some resources that I need to look for them, but I can share them. Yeah.
Mm. So I have a last question here. What would be a good system approach for disease management in like a nursery level?
Yes, so a system approach, as I mentioned, is considering all the steps and components that take place in the nursery production rather than coming with a emergency response. So, for instance, like instead of coming with a strong fungicide treatment, start looking into like what are the hazards in your nursery. So, for instance, starting like what is your source of plant material? Then what is your source of substrate? What is your source of irrigation? When once you start a propagation, where are your plants placed? Are they in direct contact with the soil? Are they above the ground? And then based on that, see how these, we call it critical control points, or these like areas in production that may pose a greater risk for pathogen infection, can be corrected. So like how can we improve the best management practices in place in order to prevent the dissemination of these pathogens.
Great. And just to remind everybody that we will be having some other Ruh Pro webinar series this year that deal with irrigation and water sanitation. So stay tuned for the lineup of this year. And I think that's all our time. Thank you all for joining us today in another wonderful session of the AFE Grow Pro webinar series. Join us next month for Dr. Raymond Cloyd presentation on quality assessment of biological control agents on February 17 at 1 p.m. Eastern Time. So please register in the link that you will find in the chat. And while in there, check out our past webinar recordings, other grower related resources, and research reports available to you for free thanks to the industry support. We ask that you please complete a brief survey about today's session where you can suggest additional topics and help us continue to improve this webinar series. Thank you again for joining us today, and thank you, Joanna, for a wonderful presentation. Have a wonderful day.